This is the second video of the ionotropic receptors lecture number 1.
So in this video, we will briefly talk about receptor pharmacology. And we'll see some basic pharmacodynamics and neuromodulation concepts and how to apply those to determine whether a drug acts as an agonist or an antagonist. So first, it's important to determine what is the difference between pharmacodynamics and pharmacokinetics, which are two very broad concepts that are applied to neuromodulation. Well, while pharmacokinetics describe the actions of what the body does to a drug, such as the influence on how the drug is liberated within the body, how it's absorbed, distributed, and processed through the metabolism to be finally excreted. Then pharmacodynamics explain or study what a drug does to the body. And we will see in the next slides where the different effects of drugs on receptors and what are the differences between different types of drugs depending on what the effects exert. So let's start with very basic concepts that involve ligand-receptor interactions. So many drugs act on sites whose specialized function is the recognition of intercellular chemical signals. That is, that they act at the recognition sites for neurotransmitter and hormones. The neurotransmitters and related drugs which interact with the receptors to stimulate the natural response are frequently termed agonists. In contrast, antagonists interact with receptors to prevent response. And drugs can be either agonist or antagonist, and we will see how we can distinguish the two of them. So now, before we get into the functions of agonist and antagonist, let's briefly talk about just the life cycle of receptors. So each cell's DNA contains genes that code for every single protein and receptor subunits. And they're synthesized from the nucleus and they produce different types of receptors that once they are made, they are assembled into the membrane. Then once assembled, receptors get embedded into the membrane and can receive and respond to signaling molecules. However, what happens if they receive too much stimulation? It could potentially damage the cells. Well, fortunately for us, cells have the ability to downregulate receptors, and meaning that they can take them out of the membrane and recycle them, which leads to fewer number of expressed receptors and therefore decreased sensitivity to signaling molecules. On the other hand, if most of cell receptors get blocked and then cells receives very weak signals from their agonist, the cells can now have the ability to upregulate the receptors and internalize more receptors into the membrane, so therefore increasing cell sensitivity to signaling molecules. So the consequence of a drug-receptor interaction is a physiological change that can actually be measured, and its magnitude can be plotted against drug concentration to produce a graded dose-response relationship. And we will see how we can interpret this normalized response, this graded dose-response relationship to determine whether a drug is acting as an agonist or as an antagonist. So now let's talk about efficacy and potency. And let's look at the example where our signaling molecule is actually some kind of drug, like drug A. So as the concentration of that drug increase its pharmacological effect. This increases until it reaches the point at which all receptors are occupied. And if we plot this, we can determine the EC50 from the graph. So EC50 is simply the concentration of a drug that produces 50% of the maximal effect. And what the EC50 tells us is how potent the drug is. So let's call this curve drug A. Now, if we can graph the dose response of a different drug, let's say drug B, we can now easily tell that between the two drugs, drug A is more potent because it has a lower EC50. Simply less drug is needed to get half of the max response. Now, what else we can see in this graph? Well, what we can see is that the drug B doesn't even reach to the same level of pharmacological effect as drug A. And this is due to its efficacy. So the maximum efficacy of each drug is represented by the Emax, at which we assume that all the receptors are occupied by the drug, and higher concentrations of it doesn't produce larger effects. So in this example, drug A is not only more potent, but it's also more efficacious. So we see here that the EC50s represent potency, while the Emax represent efficacy. So now let's talk about intrinsic activity of drugs. So intrinsic activity refers to the ability of a drug to produce a maximum biological effect. So if a drug binds to a receptor and it's able to produce a maximum effect that is comparable to the effect produced by our body's own endogenous ligand, we call it a full agonist. So now let's say that about just 15% of receptors show some kind of activity where there is no agonist around. This is what we call basal activity. So again, in the presence of a full agonist, we would see a maximal effect. If we have an agonist now that is unable to produce a maximal effect when it occupies all the receptors, we can call it a partial agonist. And lastly, if we have an agent that binds to the receptors, and instead of activating them, it stabilizes the receptors in their inactive form, we call it inverse agonist. And this is because it simply eliminates basal activity. So normally agonists enhance cellular activity, although there are some specific agonists, such as inverse agonists, that do not enhance the activity but rather stabilize the basal activity. So on the other side of the spectrum, we have antagonists, which refer to a ligand that combine to a receptor and block it, and therefore reduces agonist activity. So if we have both agonist and antagonist that can bind to the same side of the receptor, they will compete for that side. And therefore, an antagonist that can bind and prevent agonists from binding, it's called a competitive antagonist. The characteristic of competitive antagonist is that it shifts the dose-response curve of the agonist to the right. So for example, when an agonist is in the presence of a competitive antagonist, we need higher doses of that drug to get half of the maximum effect, which is this new EC50. So therefore, what is happening is that this competitive antagonist reduces the potency of an agonist. Now, some antagonists can form covalent bonds with active sites of the receptor and then irreversibly block it. This is called a non-competitive or an irreversible antagonist. And in this process, a reversible antagonist cannot be displaced by an agonist. This leads to a reduction in the maximal effect, or in Emax, and therefore results in a reduction in the agonist efficacy. So the last important concept in pharmacodynamics that I would like to talk about is the therapeutic index. Our bodies are very complex, and therefore not everyone will experience the same exact effect from the same dose of a drug. This is why we came up with the therapeutic index, as it helps us to measure the relative safety of a drug for a particular treatment. So the therapeutic index is simply the ratio of the dose of a drug that produces toxicity in 50% of the population to the dose of a drug that produces an effective response in 50% of the population. So if we were to graph and we need to determine the curve that represents positive therapeutic response and that represents the negative toxic response, then we will come up with two parameters. The ED50 that represents this concentration at which there is a desired effect in 50% of the population, and the TD50, which represents the toxicity at which 50% of the population experience a negative response. So now, having all of this in this graph, we can easily determine the therapeutic index, which is basically the range between these two concentrations. And this is how we can determine the therapeutic index of a particular drug.
And here you can find as usual a short quiz that revises some of the most important concepts from this video.
I also leave you here a few problems that we will see in class to cover in practice some of the concepts that we're seeing in this video, such as determining the efficacy and potency of a particular drug in this problem, practice how to calculate the therapeutic index of a drug, and also to determine whether a drug, depending on its dose response, it could be an agonist or an antagonist.